This is an overview of an article entitled Coagulation Factors and the Risk of Ischemic Heart Disease, a Mendelian Randomization Study, published in the January 2018 issue of Circulation, Genomic and Precision Medicine. The underlying mechanisms of acute coronary syndromes, including myocardial infarction or heart attack, is the formation of intravascular thrombus that produces an obstruction to blood flow in the coronary artery. The heart muscle downstream of this obstruction in turn becomes ischemic, that is, deprived of oxygen, and shortly thereafter begins to die. Unless vessel patency is promptly restored, cardiomyocyte loss continues and a heart attack ensues. The inciting event of thrombus formation develops following rupture or erosion of the innermost layer of the coronary artery blood vessel, which exposes extravascular factors such as tissue factor and procoagulant factors circulating within the blood vessel. In many ways, this process represents a natural response of the body's clotting system, reacting to signs of damage to the blood vessel. The clotting cascade is complex and involves multiple protein-protein interactions. Understanding the factors that contribute to this process is an important step towards understanding the body's response to plaque instability, which might in turn shed light on novel druggable endogenous targets. While experimental data do exist, there are shortcomings in the available human data to help inform these investigations. The technique of Mendelian randomization offers promise along these lines. Mendelian randomization makes use of the random allocation of genetic variants that may affect an individual's biology. Since the assortment of these variants is at least in theory random, in some ways it represents nature's randomized control trial, whereby the random allocation of genes, and in turn of individuals, to an increase or decrease in activity of a disease intermediate, can then be explored to investigate that intermediate's potential effect on disease. For example, if a genetic variant in a gene A produces a decrease in gene product A, then one could divide the population into those with versus those without such a variant in gene A. One would then have a population stratified randomly by gene product A, and could then compare risk of a disease among both groups to see if pharmacologically lowering gene product A might lower risk of disease. As the allocation of alleles is generally random, limitations such as residual confounding that challenge traditional epidemiologic observational studies should be minimized, and more reliable implications about causation can be inferred. Mendelian randomization can be carried out by looking at the effects of a single genetic variant on the gene product of interest, or the effects of multiple genetic variants can be examined through the use of a genetic risk score. However appealing the approach of Mendelian randomization might be, some important assumptions must be carefully explored first to assure that the results from Mendelian randomization can be more reliably interpreted to imply causation. First, the instrumental variable, that is, the genetic instrument used to conduct the Mendelian randomization study, must be robustly associated with its proxy. In this case, the SNP score must be robustly associated with the coagulation factor in question. Second, none of the genetic variants included in the summary score can be directly associated with any potential confounders of the relationship between the exposure and the outcome. This assumption is challenging to test empirically because some confounders can be unknown or unmeasured. Finally, the third assumption precludes any pleiotropic effects of the genetic predictors on the outcome. That is, each genetic variant included in the risk score must only affect the risk of ischemic heart disease via its association with the coagulation factor, for which it serves as the proxy. We'll see in just a moment how the authors of this particular study approached verifying these assumptions. With this background in mind, the authors of the study under discussion hypothesized that Mendelian randomization could shed light on potential determinants of increased risk of atherothrombosis in humans. They first identified previously published studies employing genome-wide association studies, or GWASs, to identify single nucleotide polymorphisms, or SNPs, associated with levels or activity of a number of important coagulation factors, including von Willebrand factor, thrombin generation potential, factors 5, 7, 8, and 10A, tissue plasminogen activator, PAI1, D-dimer, and thromboxane. In some cases, genome-wide association studies were not available. This was the case for factors 5, 7, 10A, and thromboxane. And in those cases, the authors used previously known associations in the genes responsible for the coagulation factors in question. They then constructed summary genetic risk scores for each major coagulation factor based on top SNPs from published GWAS studies. Then, they applied these summary genetic risk scores in a large case control cohort, cardiogram, to determine if the frequency of SNPs that affected levels or activity of a particular clotting factor was higher among those with or those without ischemic heart disease. But before doing the Mendelian randomization study, the authors needed to verify a few key assumptions. First, they needed to ensure that each instrumental variable was robustly associated with its proxy. For simplicity's sake, we'll just look at their results for von Willebrand factor, as shown on this slide, although they did this for each coagulation factor under study. As you can see, robust associations were present, in many cases replicated in multiple sources in the literature, strengthening the validity of these SNPs as determinants of coagulation factor levels. Second, as we mentioned, the authors needed to verify that none of the SNPs included in the summary score were directly associated with any potential confounders of the relationship between the coagulation factors and ischemic heart disease. This can be hard to do in practice, but some precautions were taken. For example, one SNP for von Willebrand factor was in the ABO gene, which has also been associated with levels of the inflammatory cytokine interleukin-6, or IL-6. Inflammation has been shown to be causally associated with ischemic heart disease, and hence the authors excluded this SNP from their analysis. Third, as we mentioned, the authors needed to verify that each SNP included in the risk score only affected the risk of ischemic heart disease via its association with the coagulation factor for which it served as the proxy. To satisfy that assumption, the authors checked for known associations between the proxy SNPs and other phenotypes, and also tested for unknown pleiotropic effects using techniques known as the weighted median and the MR-Egger method. Additionally, the Mendelian randomization analysis was extended to test for associations between genetically predicted coagulation factors and blood lipids, an intermediate phenotype on the pathway to heart attack, using data from the Global Lipids Genetics Consortium. Again, here we show the results only for von Willebrand factor, although analyses were done for all the various coagulation factors. Okay, so what did they find? Well, first, the author's most robust finding implicated endogenous thrombin potential, or ETP, as a modest causal risk factor for ischemic heart disease, though paradoxically, also as a predictor of increased high-density lipoprotein, or HDL, cholesterol. SNPs in thromboxane synthase may play a role in ischemic heart disease events. Intriguingly, thromboxane A synthase is active in the arachidonic acid metabolic pathway, the pathway which aspirin targets. While there's considerable uncertainty about the degree to which genetic factors influence aspirin's effectiveness in individual people, one wonders if some of this relationship might be explained by this possibility, although to emphasize this is very speculative at this stage. The other examined coagulation factors were inconsistently associated with ischemic heart disease. As expected, there was notable evidence of pleiotropic effects and ABO polymorphism involvement, emphasizing the importance of carefully evaluating the assumptions underlying Mendelian randomization. So is this the end of investigation into the potentially pathogenic roles of many of these coagulation factors? Well, not necessarily. Because of the difficulties inherent in validating some of the principal Mendelian randomization assumptions, these results here should be viewed as evidence, but not proof, of a lack of causal relationships between some of these clotting factors and ischemic heart disease. For example, the authors found that variation in the gene coding for factor 10A did not seem to increase the risk of ischemic heart disease events. In contrast, a very recent large randomized trial, the COMPASS trial, suggested that the addition of rivaroxaban, an inhibitor of factor 10A, to standard therapy in patients with a history of vascular disease, reduced the composite of cardiovascular death, stroke, or myocardial infarction by 24%. For a number of reasons, these seemingly discordant findings may not be in conflict. However, at the very least, it bears mentioning that there may be specific populations or specific clinical contexts in which some of these clotting factors have more of an effect on ischemic heart disease. In conclusion, Mendelian randomization can help establish a potential causal relationship between risk factors and disease, potentially avoiding some of the residual confounding problems that observational epidemiologic studies face. The findings here implicate endogenous thrombin potential as a causal risk factor for ischemic heart disease, as well as more broadly emphasize the connectivity between coagulation and lipid risk factors for ischemic heart disease. Overall, these results provide an important evidence base for the design and planning of future studies of coagulation pathways and heart attack.